Okay, hello everyone. I'm Nicolette Cook, and today I'm going to be talking about blood vessels. And before we get into any of the information, I'm going to be going over some of the learning objectives for this lesson. Our first learning objective is to compare and contrast the three tunics that make up the walls of most blood vessels. 2, we're going to distinguish between elastic arteries, muscular arteries, and arterioles on the basis of structure, location, and function. 3, we're going to compare and contrast arteries and veins. 4, we're going to describe the basic structure of a capillary bed from the supplying meta-arteriole to the venule into which it drains, and describe the different types of capillaries. 5, we're going to explain the structure and function of venous valves into the large veins of the extremities. And 6, we're going to discuss several factors affecting blood flow into the venous system. Okay, topic 1, we're going to get started with blood vessel structure. Blood vessels are structures that carry blood throughout the body. They play a huge role in the thermoregulation and controlling of blood pressure. But obviously their biggest function is to transport blood. We then have arteries, and then they will carry blood away from the heart. And we will get into their function later on in this lesson. They then branch into arterioles, which deliver blood into the capillaries, where nutrients and waste will be exchanged. Capillaries then carry blood to venules, which carry blood to a vein, which then returns the blood to the heart. With blood flowing through these vessels, there has to be a hollow passageway, which is known as a lumen. And this shows vessels that transport oxygenated and deoxygenated blood, this figure right here. And we are going to continue on our first topic, and we're going to be getting into tunics. Continuing with blood vessel structure, we're going to be talking about the three tunics. They make up the walls of arteries and veins, and there are 3 of them: the tunica intima, tunica media, and the tunica externa. We're going to start with the tunica intima. The tunica intima is the innermost layer, and it comes in direct contact with blood. It is made up of epithelial and connective tissue. The epithelium is called the endothelium and is continuous throughout the entire vascular system. This releases local chemicals called endothelins that can constrict the smooth muscle within the walls of the vessels in order to increase blood pressure. Beside the endothelium is the basement membrane, which we can see right here, which binds the endothelium to the connective tissue. It is permeable and allows for material to pass through. In larger arteries, there is an internal elastic membrane, and it provides structure, but also allows a vessel to stretch. This is not in veins. Again, the internal elastic membrane will not be found in veins. Next is the tunica media, which is right here on this structure, which is the middle layer of arteries and veins and is much thicker in arteries than in veins. It is actually the thickest layer in arteries. Contraction and relaxation of the smooth muscle causes the size of the lumen, again, which is the opening, to decrease or increase, causing vasoconstriction, which decreases blood flow, and vasodilation, which increases blood flow. Both are regulated by small vascular nerves known as nervi vasorum, and they run in the walls of blood vessels. The external elastic membrane is also found in the tunica media, and it is not normally seen in smaller arteries nor veins. Finally is the tunica externa. This is the outermost tunic, which is made of connective tissue. In veins, it contains groups of smooth muscle fibers as well. It is also the thickest tunic in veins as well. The tunica externa anchors blood vessels to surrounding tissues, and embedded in the tunica externa is the vasa vasorum, which is a collection of small blood vessels that brings required nutrients and oxygen to vessel walls. And right here is the tunica externa. All right, we are moving on to topic 2, which is we are going to be talking about elastic arteries, muscular arteries, and arterioles. Elastic arteries, which can be found right here, are closest to the heart. They have their name due to them containing the highest percentage of elastic fibers in their tunica media, which can be seen here as like the thicker section of the elastic artery. Their vessel diameter is normally larger than 10 millimeters. Since they have so many elastic fibers, these arteries expand as blood is pumped from the ventricles and passes through them, and they recoil after the surge of blood is complete. And the elastic recoil helps to maintain the pressure gradient that allows blood to flow through these arteries. Next, we're going to be moving right along to muscular arteries, and they are further away from the heart, where the rush of blood is more slowed down and relaxed. And they branch off of elastic arteries, and their vessel diameter could range from 0.1 millimeters to 10 millimeters. They have a thick tunica media, as shown right here, that allows for muscular arteries to play a leading role in vasoconstriction, which, as we talked about before, decreases blood flow. And they also funnel blood into the smallest diameter artery known as an arteriole. And that's where we get into the arterioles. They have vessel diameter of 30 micrometers or less. And arterioles lead to capillaries, and they play a huge role in the slowing down of blood flow. They are referred to as resistance vessels because they resist blood flow and often cause a huge drop in blood pressure. One cool thing about arterioles is that they are the primary site of both resistance and regulation of blood pressure, and this occurs by them regulating their vessel diameter. And this is controlled by neural and chemical controls, and vasoconstriction and vasodilation in arterioles are the primary mechanisms for distribution of blood flow into capillary beds. So as we can see, the tunica intima on the arterioles also very wavy in structure. And the tunica media is very thick, where the tunica externa is very thin. And that's just another picture of the three. And now we're going to be moving on to topic 3. We did talk a bit about veins and arteries in the beginning of this, but now we're going to be getting into the difference between the two. To begin, veins return blood to the heart, as opposed to arteries, which carry blood away from the heart. Veins also have very thin walls, and their lumens or openings are large and will appear flat, whereas arteries they have thick walls and small lumens that will look rounded. The tunics in veins and arteries differ as well, and arteries in the tunica intima. The endothelium is wavy, as we can see right here. And the elastic membrane is present, whereas in the veins, the endothelium appears smooth, as shown right here, and the internal elastic membrane is not present. In arteries, the tunica media is the thickest layer, and smooth muscle cells and elastic fibers are predominant, and the nervi vasorum is present. In veins, it is thinner, and smooth muscle cells and collagenous fibers are predominant, and the nervi vasorum is present as well. For the tunica externa, in arteries, it is thinner, and the nervi vasorum and vasa vasorum are present, whereas in veins, it is the thickest, and both the nervi and vasa vasorum are present as well. Now we're going to be moving on to our 4th topic, which is capillaries. They are normally only one cell layer thick and consist of only an endothelial layer surrounded by a basement membrane. The diameter ranges from around 5 to 10 micrometers, and capillaries are the site for gas exchange, and exchange of gases and other substances occur in capillaries between blood surrounding cells through perfusion, and the flow through capillaries is called microcirculation. So we can kind of see those endothelial cells in here in that basement membrane around that capillary. And these are just some other, we'll be seeing some of these terms later on in the presentation. And now we're going to continue on the topic of capillaries and move on to capillary beds. Now, blood flow into capillary beds is controlled by a meta-arteriole. And this is a type of vessel that has characteristics of an arteriole and capillary. Each meta-arteriole comes from a terminal arteriole and then eventually branches off in order to supply blood to a capillary bed that could have from 10 to 100 capillaries. Now we will bring up precapillary sphincters. They are circular smooth muscle cells that tightly regulate the flow of blood from meta-arterioles to the capillaries they supply. They funnel blood and oxygen to tissues where it is needed. And blood flows from a meta-arteriole into a thoroughfare channel and then into the venous circulation, and this creates a vascular shunt. And blood flow through these capillaries is irregular and is called vasomotion, and this is regulated by chemical signals that are normally triggered in response to changes in internal conditions. And so this circular portion right here is the capillary bed, and you can see just how many branches it has going off of it as well. And we're going to continue on the topic of capillaries again, but we're going to be talking about the 3 different types of capillaries, and they go in order from least leaky to the most leaky. So we have continuous, fenestrated, and sinusoid capillaries. And continuous capillaries are the most common type. They are found in almost all vascularized tissue. They are characterized by a complete and relatively impermeable endothelial lining. In continuous capillaries, endothelial cells are held together with tight junctions and just a few intercellular clefts, as we can see right here on the image. And these clefts allow for exchange of water and other very small molecules. Again, leakiness in this type of capillary is not the leakiest type, and the only substances that can pass through are metabolic products. So this could be glucose, water, gases, hormones, and white blood cells. Next is fenestrated capillaries. They have pores within their endothelial lining, and they make the capillary permeable to larger molecules. And these capillaries are found wherever rapid transport is required. They're common in the small intestine, the kidneys, and in the choroid plexus of the brain. So we can see those pores right here. They're very small, but as you can see in the continuous, there are no pores, whereas fenestrated, we are starting to get those porous openings. Finally is the sinusoid capillary, and they are the least common and the leakiest type of capillary. They have extensive intercellular gaps and incomplete basement membranes, as well as intercellular clefts and fenestrations, which again are those pore-like openings. And as you can see by the image, it almost looks like Swiss cheese, kind of, with how big the openings are. That's kind of how I've learned to remember it. And these pores allow for passage of the largest molecules, including plasma proteins and even cells. Blood flow is slow, which allows for more time for gas exchange, nutrients, and waste to come through. They are found in the liver, spleen, bone marrow, lymph nodes, and many endocrine glands. And as we can see, the basement membrane is incomplete as well, which allows for it to be extremely leaky, and blood flow is going to be extremely, extremely slow. Now we're going to be moving on to topic 5, which talks about venules, veins, and venous valves. Now, venules are the smallest type of vein, and they are normally 8 to 100 micrometers in diameter. They drain and exit from a capillary bed, and multiple venules join to form medium-sized veins, which is this image right here. And here's the image of a venule. We've talked about veins a little bit before, but just as a refresher, they do carry blood towards the heart. They are thin-walled with large lumens, which are the openings. And since they are under low pressure, larger veins will have valves that promote the unidirectional flow of blood towards the heart and prevent backflow towards these capillaries. So we can see these valves are going to stay closed here, right? And they will have 2 pumps to increase pressure. First is the skeletal muscle pump, which can be shown right here. And it helps to lower pressure. It helps the lower pressure veins counteract the force of gravity by increasing pressure to move blood back to the heart. And the contraction of skeletal muscles that surrounds a vein increases pressure in that area, which then forces blood closer to the heart. And then next we have the respiratory pump, which I do not have pictured here, sadly. It aids in returning blood from the veins to the heart, and it's driven by pressure changes in the both thoracic and abdominopelvic cavities during inhalation. And during inhalation, the venous pressure in the thoracic cavity increases while venous pressure in the abdominopelvic cavity decreases. And then that pressure difference promotes movement of blood towards the heart as well. So we can see all veins and venules and venous valves are all going to be helping to push blood towards the heart, which we can always associate veins with. We're now moving on to our sixth and final topic, which is blood flow in the venous system. And although veins return blood to the heart, they can also serve as blood reservoirs. And within systemic veins, which we can see right here on this blood distribution chart we have going on over here, 64% of our blood is stored in those systemic veins, with capillaries having 5%, the heart having 8%, pulmonary vessels 12%, and systemic arteries holding 15%. And this is due to high capacitance, which is the capacity to expand when needed to store a high volume of blood. And when blood has to be redistributed, the vasomotor center in the medulla oblongata sends sympathetic stimulation to the smooth muscle walls of the veins, which then causes constriction. This increases pressure on the blood within the veins, causing a speedy return to the heart. And sometimes we see like a slowdown. This could be caused due to the cardiovascular system and could lead to edema, which is swelling in the hands due to a fluid buildup. And then we just have our citations.